Hello there, welcome to your first fully vodcasted lecture this semester. If you're joining me from the regularly scheduled classroom, then this is Friday, and you're looking towards a couple of up-and-coming events. First off, homework 5 will be due on Wednesday rather than Tuesday, and I've done that because your practical exam is going to be on Tuesday, October the 15th. So you'll want to review for that practical exam, and hopefully you found your way to the review that is posted on the lab website. You'll also find the virtual edge to be incredibly helpful in studying for the practical. Go back and watch the videos of T-streaking and aseptic pipetting because these will really help you on the hands-on stations on that exam, one of which will certainly be a station in which we'll be watching your aseptic technique while pipetting, and one will be a T-streaking station. So you're going to want to be ready and able to do those. You might also want to watch the video on the microscope and thinking back on how to focus your microscope, because that is going to also be one of the hands-on stations there. So just a few things that are up and coming, and if you have not had a chance yet to finalize your poster presentation hypothesis with me, please try to do that as soon as is possible for your group. When we left off last time, we had just been talking about the way in which a cell can metabolize fat, and we had looked at the energetic nature of that process. We hadn't yet gotten to the protein as a fuel and the way in which proteins can in fact enter into catabolism. So this is our last piece of coverage for chemoheterotrophic catabolism, and perhaps it is fitting to end with the least favored fuel of the cell. So while proteins can be used in catabolism, they have to actually be detoxified before they can be used. That's why we say that in fact protein is a bit of a toxic fuel, because organisms that do incorporate it into catabolism have to first detoxify or deaminate the amino acids that will enter in. So in this picture, you can see that I'm trying to show you a polypeptide chain from amino terminus to carboxyl terminus, showing each one of the amino acids as just a little ball. But if we further deconstruct and say that perhaps, in fact, this first amino acid is the amino acid glutamate, you can see it here. And if that is liberated after the breaking of a peptide bond, then what we notice is that this amino acid is now pretty much the same as one of the intermediates in the TCA cycle. So in the same way that alpha-ketoglutarate could be used as a precursor metabolite in the reverse reaction to make glutamate, we can see the opposite thing happen, where glutamate can literally become deaminated to then form alpha-ketoglutarate. So the ammonium right here has to be removed from, so the amino group there has to be removed from the carbon skeleton of the glutamate. And then that carbon skeleton can enter into TCA cycle as alpha-ketoglutarate. So while proteins are a possible fuel source, the fact that we have to remove that amino group makes them a less preferred fuel. And you might recognize that in humans, we secrete the amino, the leftovers, in a cycle called the urea cycle, and urine is a product of that. So urea is the primary component in urine. Many other organisms secrete this in other ways. But we recognize then that proteins can, in fact, enter in to catabolism when enzymes called proteases, of course, break those peptide bonds. And then deamination takes place to allow these amino acids to enter into catabolism. And depending upon the amino acid, they'll enter in at slightly different places. For example, the amino acid alanine, it can enter in as pyruvate. It's closer in structure to that catabolic intermediate. So it's a bit sad to say that this is where we leave chemoheterotrophic metabolism completely behind, but you'll be super stoked to know that we're now going to jump into one of my favorite topics. And this is our chemolithoautotrophs. Some of you will recall that lithotroph quite literally means rock eater. So no matter what I do, every time I think of a lithotroph, I can't help but picture our good friend from the Never-Ending Story, who is the rock eater. So I brought in a picture of the rock eater, and one of my students actually previously made an awesome cartoon that I'll show you in lab that just says chemolithotrophs rock. So we get the idea that they're taking in inorganic chemicals. And in fact, prokaryotes are actually alone, although we have the mythical rock eater that is definitely not prokaryotic, in our known terrestrial environment, we recognize that there is really no such thing as that fictional character. And in fact, it is only the prokaryotes that are capable of utilizing rocks as their source of energy. Said another way, that is inorganic chemicals. And so while rocks would be one good example of that, we could also think about inorganic chemicals as deriving from other sources. And one of those that you might readily think about is, in fact, byproducts of anaerobic respiration. So remember, we just talked about anaerobic respiration, in which an organism, even E. coli, can use something like nitrate as its terminal electron acceptor. And when it does so, it reduces that compound and it makes something that often looks like dinner to a rock eater. So many rock eaters will live happily in environments where there are many anaerobically respiring bacteria that do produce the things that they like to eat for dinner. Good examples of those would be those that reduce sulfate, making hydrogen sulfide, those that reduce nitrate, making nitrite. They're actually very nicely suited to live in an environment along with a chemolithoautotrope. One man's trash is certainly another man's treasure. Let's look specifically at some of these chemolithoautotrophs, beginning with the hydrogen bacteria. Imagine looking at hydrogen gas and seeing that as your dinner. These bacteria will rip and strip high energy electrons off of hydrogen gas. There are others that see sulfur as their source of energy and will in turn rip and strip high energy electrons off of hydrogen sulfide. And in many cases, they will actually create sulfuric acid as a byproduct of this catabolism. So recognizing the possibility of some of the byproducts of chemoautotrophs as being things that are very likely to solubilize minerals. So we're going to come back to that momentarily. Keep it in mind. We also have the iron bacteria, and iron bacteria are going to take iron 2 plus and convert it into iron 3 plus. That is oxidizing or removing electrons from these ions. So Ethan will remember our conversation just the other day in Open Lab and recall that we were talking about how oxidation can sort of take different forms, but we mentioned that with metal ions, oftentimes that's going from the 2 plus form and to the 3 plus form, so taking an electron from that. And that's what iron bacteria do. They rip and strip an electron from the ferrous form of iron. Now, our nitrifying bacteria come in two different types. There is the type that likes to consume and eat nitrite, oxidizing nitrite to nitrate. And then there's the type that takes ammonia and converts ammonia eventually to nitrite. And you might be getting the sense that nitrifying bacteria play an incredibly important role in the nitrogen cycle, right? They're absolutely essential in the portion of the cycle that we call nitrification. And so super, super awesome role that they play environmentally. Now, one thing to note is that while rock eaters are very different from us in what they see as their source of donuts, we could also say that they hold some very great similarities with us in that they have a fully functional electron transport chain, and that electron transport chain is often using the same terminal electron acceptor that we use. That is to say, these are often aerobic in their ETC utilizing a terminal electron acceptor that's the same as that our mitochondrial ETC uses. So it's just that what they rip and strip their electrons off of is very, very different than what we like to eat. But certainly they hold some similarities in how they then use those electrons to make energy and gradients and eventually ATP. We might guess that chemolithoautotrophs thrive in very extreme environments where they tend to find their reduced inorganic compounds. Very extreme environments where maybe there are also a lot of anaerobically respiring bacteria producing the waste products, so to speak, that then chemoautotrophs see as a source of energy. So while we might say that there's not a whole lot of high energy capacity in something like a hydrogen molecule, what we can also note is there's not a whole lot of competition for that hydrogen molecule either. Not everything is vying for that and fighting over that particular donut. So these can be they can be less efficient in their energy generation because they don't have much competition for that particular energy source. I want to talk specifically about a particular chemoautotroph that is very close to my heart, and that is Thiobacillus ferrooxidans. So Thiobacillus ferrooxidans is notorious in acid mine drainage. So what happens when mining takes place is that there's a lot of upheaval of the minerals, and there's fractures and things, and this what happens is that this exposes iron pyrite or iron sulfide. So, iron sulfide, also iron pyrite, and if anyone else grew up in a mining town, then you will also realize that iron pyrite equals fool's gold. And fool's gold, pretty cool stuff, actually. We used to have an awesome doorstop. We had a huge chunk of fool's gold. It was an awesome doorstop. And it's cool to think about a bacterium that would like to eat that for dinner. But what's even more interesting is to think about in a community with a lot of mining taking place, that what happens there is that, again, the fractures and upheavals expose the iron pyrite to the oxidizing zone, which means that bacteria like Thiobacillus ferrooxidans. So let me jot down the name of this bacterium that oxidizes iron sulfide. Its name is thio, which some of you will recognize that relating to sulfur, bacillus ferrooxidans. And so what we recognize from that awesome name is that this is a bacterium that oxidizes both sulfide and iron. So when there is exposed regions to this oxidizing zone, we get the conversion of fool's gold into sulfuric acid. So you can see the sulfate ion forming along with acid there. So that'll become very rapidly, can form sulfuric acid. We also recognize, though, that this iron 2 plus that is formed when that first reaction takes place can further become oxidized, forming the iron 3 plus species. And so we can get the further oxidation of that iron as well. So Thiobacillus ferrooxidans oxidizing both of these as its donuts. And what happens with that is that we get acid mine drainage. So we might just note acid mine drainage is a big deal. And so this is a picture from just outside of my hometown. This is on the bike path that is actually just east of where I live. So what has happened here is a lot of acid mine drainage where production of sulfuric acid due to Thiobacillus ferrooxidans and its oxidizing and its whole chemolithoautotrophic behavior has led to a lot of this really, really orange colored runoff because of the sulfuric acid and the solubilizing of minerals that occurs due to that decreased pH, that acidity. Let's now move on to something that Hannah and John are going to be very excited about with your passion for plants. The idea of looking at photosynthesis should resonate with you. So phototrophs, arguably the most important organisms in our environment. They are the primary producers of our environment. That is, they capture light energy and convert it into chemical energy. So the capture and conversion of one form of energy into another form of energy, displaying in a nutshell the first law of thermodynamics. So we could say that photosynthesis is the backbone of all life, because certainly in our terrestrial environment that's true, although we shouldn't limit out chemoautotrophs because we know that they too are very much important in being primary producers in environments that lack sunshine. But we recognize in ours that does have sunshine that these are the primary producers, and that they really literally are taking our byproducts, right, and converting them into a very usable carbon source, an organic carbon source. So let's look at the balanced reaction for photosynthesis. And I know all of you would tell me this part, right? It's the making of sugar or the creation of glucose from carbon dioxide. So building from the precursor metabolite of CO2 and synthesizing sugar. And we recognize that as being the case. We know that in order to do this, plants need light, but of course what Hannah will also say is plants also need water, right? Well, that's true, and we're going to get there, but it turns out that not all photosynthesis is the kind of photosynthesis that uses water as the electron donor. So instead of making that statement, I'm going to make a much broader statement to say that in photosynthesis, we always need an electron donor. So we need some molecule, whether that be H2O or whether that be H2X. Maybe it could be something like hydrogen sulfide instead of water that would be serving as the electron donor. So if it is water, we know then that the byproduct of the oxidation of that water is oxygen gas. And that is true for plants and for other oxygenic phototrophs. So if O2, then oxygenic photosynthesis. And said another way, if the electron donor is water, then we're looking at oxygenic photosynthesis. But not all photosynthesis is oxygenic, that is oxygen creating, oxygen producing. Some photosynthesis will produce something besides oxygen, and that's why we're using that broad X character there. We could see that in fact sometimes that molecule is sulfur or some oxidized form of sulfur. So it depends upon what type of photosynthesis we're looking at. And in order to better understand that, I want to take a look at a Winogradsky column that turned out really cool. This shows a lot of the nice layers that we get in a Winogradsky as they develop. We can see down in the very bottom a lot of anaerobically respiring and or just overarchingly fermentative bacteria. Then we get these lines right here, and these lines here are anoxygenic phototrophs. That is, they don't use water as their source of electrons, but they do use something else. So anoxygenic phototrophs, and in fact, specifically, these are green sulfur and purple sulfur bacteria, and they utilize hydrogen sulfide as an electron donor. So H2S is donor here. Whereas up above in the top area, we see a lot of oxygenic phototrophs. And these oxygenic phototrophs do use water as their source of electrons and therefore do generate oxygen. So let's take a quick look at the two types or the two reactions into which we can split photosynthesis. On the left, the light-dependent reactions. On the right, the light-independent or dark reactions. The light-dependent reactions, of course, by sheer virtue of definition, are those that require light in order to harness the energy of that light and convert it into chemical energy in the form of ATP. Said another way, the sunlight excites electrons, and those electrons pass through an ETC. That ETC pumps protons, and that proton motive force is tapped into using ATP synthase to make ATP. And that ATP can then be used to power the dark reactions of photosynthesis. That is, more explicitly, the building from carbon dioxide up to organic carbon. This is quite often called carbon fixation. Now, the light-dependent reactions also yield something besides ATP. That is, we know that in order to power a biosynthetic reaction, which is very much what the light-independent reactions are, we know that reducing power is also needed. And those of you who really have your sexy minds on will recall that the reducing power needed is NADPH, right? The stuff that's pumped into biosyntheses. So from where does that derive? Well, we know two different places depending on whether it's an oxygenic or anoxygenic phototroph. If it's oxygenic, then we know that it comes from water, and electrons are stripped off of water to create reducing power, and of course that derives oxygen as a byproduct. We could put in parentheses here that this particular reducing power is NADPH. We know so, right? Because that's the type of reducing power that's used in biosynthesis. So that's going to be the type of reducing power utilized in the light independent or dark reactions. Oxygenic photosynthesis, well, yes, it is the one that's done by plants, but remember that more than half of the world's oxygen is generated from microbial life. So it's not just plants, but it's also algae and of course our prokaryotes such as cyanobacteria that are also capable of ripping electrons off of water. But of course we know that oxygenic photosynthesis is not the only kind, and not everything strips its electrons from water. So we should look at the anoxygenic phototrophs as well. They get their reducing power, their NADPH, from hydrogen sulfide, for example. That's a good example of anoxygenic photosynthesis. And that's what's used by those really beautiful purple and green sulfur bacteria. They are ripping and stripping their electrons off of H2S. Now you might be thinking, Rachel, why the heck did you put this down in the middle of no man's land? I mean, is it light dependent or is it what, not light dependent? The reason that I did this is because, yes, it is light dependent, but the light is IR light. So that is to say it's very shifted to long wavelengths of light that are not particularly visible. So we can now make a broad statement that although the light independent reactions don't require light, they are utilizing the products of the light dependent reactions in phototrophs. So we recognize that the products of the light reactions are really needed to power the dark reactions. And this is what we see here. On the upper panels, we see the most amazing organisms on Earth, my absolute favorite group of bacteria called the cyanobacteria. Now down here, we get to see another cyanobacterium, and over on the right, this is a picture of purple sulfur bacteria. So these are oxygenic phototrophs, right? Top panel left here, oxygenic phototrophs. Anoxygenic phototroph on the lower right. And remember that the anoxygenic phototrophs are very different than oxygenic phototrophs because they absorb different wavelengths of light. Let's take a quick look at that. We know that all photosynthetic organisms have light-absorbing pigments. Some of those pigments include chlorophylls, carotenoids, and phycobiolins, and all of these are colored so as to capture light energy. They, of course, vary in color depending upon the wavelength of light that they absorb. And depending upon if an organism expresses a lot of chlorophyll, then it may not be possible to see as much of the pigmentation from the carotenoids and phycobiolins. But if, like the purple sulfur bacteria, that they don't produce chlorophyll, then we can see a lot of pigmentation coming from the accessory pigments. So let's make note that the oxygenic phototrophs, like cyanobacteria, have chlorophyll A and B. And chlorophyll A and B absorb light at about 440 and 660 nanometers, meaning that they absorb blue and red light, leaving the green light to be what hits our eyes. So we would recognize the cyanobacteria of having green colors, and that was what's true on the earlier slide that we just looked at. They were green. Whereas when we think of the purple and green bacteria, we recognize them as not having any chlorophyll A and B, but instead having an alternative primary pigment called bacterial chlorophyll. Bacterial chlorophyll absorbs in the IR region of, 900 to around 1,000 and something. So they absorb at a slightly different wavelength. Now, what is true about that is that green and purple anoxygenic phototrophs never compete with oxygenic phototrophs for ecological niche. We might ask, well, what is different here in these bacterial chlorophylls versus chlorophyll A and B? Well, one thing is that chlorophyll A can extract electrons from water, right? And we know that to be true, it's a strong enough oxidizing reagent to extract electrons from water. So these are things with chlorophyll A and B. These are oxygenic phototrophs, right? Whereas those with bacterial chlorophyll, these are our anoxygenic ones. They are not strong enough oxidizing. So bacterial chlorophyll is not a strong enough oxidizing agent to extract electrons from water. Instead, it extracts electrons from H2S. Carotenoids and phycobilins, these are the accessory pigments. That is, they exist in concert with chlorophyll A and B or bacterial chlorophyll and help to enhance the amounts of wavelengths of light that can be absorbed. They can be very photoprotective, but also they can give brilliant colors to whatever it is that it contains those pigments. So with the green and purple bacteria, that's actually the accessory pigments that we're seeing. So you might ask, well, are these pigments just like a mishmash, like hanging out in the middle of the cytosol of a cell, like, just in the middle of cyanobacterial cytosol? No, they don't. They're organized, very highly organized into complexes that are called photosystems. And that's where we're going to go next, is looking at these photosystems.